everybody, I'm Tall.
And I'm Small.
And today we are doing MCAT from Khan Academy. Biomolecules question. I'm totally botching this intro, but whatever. Gene control questions. We're going to talk about controlling genes. First question, what is an operon? Choose one answer. So the available answers are a unit of DNA containing a cluster of genes under control of a promoter and a repressor, a protein that binds to an RNA segment and blocks the attachment of DNA polymerase, halting transcription, a protein around which DNA is coiled until it is transcribed, also the chief protein component of chromatin, or a reversibly bound protein that enhances the transcription of a particular segment of RNA. An operon. So I'm pretty sure that that would be a unit of DNA, right?
Mm-hmm.
And that would be a unit of DNA. And I knew operon had to do with operating the gene, promoting and repressing the gene.
Yeah.
So it's a unit of DNA that that has all of the... Well, it has the promoters, or it's under control of the promoters?
It's under control of either promoters or repressors. And so the two examples that they always want you to learn for your intro bio or genetics classes are the Lac Operon and the TRP, the tryptophan operon. And they are controlled in a reverse way of each other. But they are a set of genes that are involved in an element of metabolism for one unit or another.
Okay, so the operon is like one segment of DNA, and then you can either bind a promoter or bind a repressor to that operon.
Yeah, and they're also so one of them could be like naturally on all the time and they can get turned off, or they can be like naturally off all the time and get turned on.
Oh, okay. But the operon is like the umbrella term for it.
Yeah. So like the one in the Lac Operon has I think 3 different genes. I think like X, Y, and Z maybe, and yeah. And so they all have different functions, but it's all under lactose metabolism.
Okay. All right. So we are choosing a unit of DNA containing a cluster of genes under control of a promoter and repressor, and it was right. Awesome. All right. Question number 2. In a bacteria possessing the Lac Operon, which of the following occurs when glucose is low and lactose is abundant? So this is a bacteria possessing a Lac Operon. We have low glucose, high lactose. So is it beta-galactosidase acetylases the operon? That is a weird word. Lactose permease cleaves lactose to glucose and galactose. Lactose metabolism is increased by lactose binding to the operon. Transport of lactose into the cell is enhanced. Okay, so real quick, do you know the answer to this?
I think I do, yes.
Because I think I don't. And I'm going to try to reason my way through it and tell me where I'm right or wrong.
Yeah.
So beta-galactosidase acetylases the operon. That doesn't seem to make any sense.
Okay.
Putting an acetyl group on DNA.
Also keep in mind, yes, so you're on the right track.
Okay.
And just keep in your mind that beta-galactosidase is one of our little marker things we can do with that operon in some of like the genetics labs.
Yeah, I remember that.
So it's one of the genes in the operon. So that doesn't make any sense.
Okay.
That the product of one of those genes was already affecting it if we don't know if it's getting produced or not.
Right, okay. Okay, so then lactose permease cleaves lactose to glucose and galactose. So that is that's what lactose is made of, right? It's a disaccharide of glucose and galactose.
Yep.
However, they're talking about when you have the Lac Operon, which of the following occurs when glucose is low and lactose is abundant? Does it break lactose into glucose and galactose? Yeah, I would assume, right? Because glucose would still be the most sought after energy source.
Yeah, it's definitely preferred over lactose, and that's why they specified that glucose is low, or we wouldn't be having this conversation, and the cell would just use glucose.
Right.
So don't rule that one out because it has the right concept maybe, and I don't even remember what lactose permease is. It might be coming into the cell or something, but I'm not sure, so
okay.
I can't rule it out.
Okay. The next one, lactose metabolism is increased by lactose binding to the operon. Well, we've got a lot of lactose.
Yeah, and so I think here what might get me in trouble is that, you know, because we have some genetics background research,
Mm-hmm.
I know that it's a kind of a form of lactose called allolactose that turns off the repressor and allows this operon to start being expressed.
Okay.
And so I don't know if they want us to differentiate between allolactose and lactose. But I would have gone with that one because that to me is how that operon gets turned on.
Okay. And then just for integrity's sake, I think we can get rid of transport of lactose into the cell as enhanced. Doesn't
Yeah,
seem to make any sense.
we're already working with the DNA.
So I'll go with you. I'm kind of stuck between either cleaves lactose or that the metabolism is increased. So we're gonna check metabolism is increased.
Fingers crossed.
Fingers crossed. No, not quite yet. Try
Oh
again, get help, or move
no.
on. No, thank you, Sal. We've got all the help we need. Okay, let's see if it is the other one, though. No, it's not.
Oh no.
What could it... It's the one that we ruled out. Okay, so the correct answer is transport of lactose into the cell is enhanced. Let's look at
Huh.
those hints. Okay, so the first hint, the Lac Operon contains genes necessary to enhance lactose metabolism. Okay. Cleavage of lactose is carried out by the beta-galactosidase encoded for by LacZ, not lactose permease.
Okay.
Wait,
Yeah.
so cleavage of lactose is carried out by beta-galactosidase,
Right.
not lactose permease.
Right. And I think when we have when we have this engineered into um what is the word? I'm like a plasmid when we
Mm.
yeah. The cleavage of lactose changes the color of the colony, I believe.
Turns blue, right?
Yeah. And so I should have remembered that that is what does the cleaving. And I think lactose permease is what helps bring it into the cell. And so I guess that does make sense. If we realize that we don't have glucose, which is our default sugar of choice, and lactose is present and it's something that microbe can use as a food source, then we would turn on this operon, and then it would make sense for us to bring lactose into the cell.
Okay. And yeah, when glucose is low but lactose is readily available, the Lac Operon is activated, enhancing transport of lactose into the cell as well as lactose utilization. All right. Okay. Next question. Question number 3. The effect of allolactose, so you talked about this,
Yeah.
is dissociation of RNA polymerase from DNA, terminating transcription of genes regulated by the Lac Operon, enhancement of binding of RNA polymerase to DNA, increasing gene transcription, conformational change in the repressor protein, including the Lac Operon, or repression of the Lac Operon when glucose is present. Okay, so you nodded your head for the third one.
Yeah, so to me, I always think of the Lac Operon as being off because lactose is not as, like, if I was a microbe, my sugar of choice is not lactose, so I keep that pathway off if I don't have to expend energy to use it.
Right.
So, and allolactose I know is what activates, you know, if it's off all the time, there's a repressor bound to it, and allolactose is what helps us get that repressor off.
Okay, so yeah, it would cause a conformational change in the repressor protein, inducing the Lac Operon. Okay, so that's a repressor on the operon that is stopping DNA polymerase from like getting on to the gene or anything like that. Okay, all right. So let's check it. Hey, we were right. Nice work. Nice work. Okay, next question. Question number 4. Deacetylation of histones has which of the following effects? Uncoiling of histone structure, allowing it to be accessed by transcriptional machinery. Uncoiling of histone structure, preventing it from being accessed by transcriptional machinery. Coiling of the histone structure, preventing it from being accessed by transcriptional machinery. Or coiling of the histone structure, allowing it to be accessed by transcriptional machinery. So, when we deacetylate histones, are we coiling them or uncoiling them?
You remember?
I think
Okay.
we're uncoiling them.
So
I'm wrong.
So we know our DNA is negatively charged, and we know our histones has things like lysine and arginine, I think, are the two amino acids that are most prevalent on that outside structure of the histones. And so that positive charge from those basic amino acids is what helps us bind the DNA so tightly and wind it so tightly that we can condense it. So what puts on the acetyl groups is a histone acetyltransferase, and so that gets bound to the arginine and lysine residues on the histones, and that uncoils the DNA. And so we call the HATs the helpful librarian that wants to throw all the books around she needs to in order to find you the right thing. In order to access the DNA, she'll mess up the structure however she has to. But then at the end of the day, you have the older librarians that like their structure and order, and they come put things back together. And they're older, they got a little bit longer of a name. It's a histone deacetylase or something like that. So your HDAC is the old librarian that comes to take off the acetyl groups and recoils the DNA and puts everything back where it's supposed to go.
Okay, so acetylation is going to open up histones, meaning the opposite. Deacetylation would recoil them. Mm-hmm. Okay, so now we're at either preventing it from being accessed or allowing it to be accessed. And based on just common sense almost, and what you just said, if you coil the histone structure by deacetylating it, you would be preventing it from being accessed.
Yep.
By
Perfect.
transcriptional machinery. All right, so let's check that. Got it. Awesome. Okie dokie. Question number 5. Methylation of CpG islands is essential to which process? Reversible inactivation of genes as cells terminally differentiate, enhancing cell differentiation into different organ systems with distinct functions, enhancing oncogenic potential of rapidly differentiating cells, or stable silencing of DNA as cells terminally differentiate. CpG islands. Do you even know what CpG islands are?
I don't, but I think I remember talking about methylation in a couple of different areas. I know that we use them, that SAM enzyme, or SAMe as you guys called it.
Oh yeah.
You know, to
acetyldenosylmethionine.
do the 5' cap. And then I think some other methylation was to conserve some regions that were important on the DNA. And so I would have to have made a guess on this one.
Okay. As I'm looking through it, methylation. So when you methylate DNA, that turns it on, right?
All I remember is that you're protecting it from degradation when you methylate it.
Okay.
So in the sections that we don't want to cut out when we're doing all sorts of splicing, that section is probably methylated.
Okay, so I see you picked enhancing cell differentiation into different organ systems.
Yeah, for some reason I think that also has to do with methylation, but I could be totally wrong. This is a guess.
Okay, well let's try to. I like your guess. I literally have no guess.
Okay.
But in true MCAT fashion, I'm just gonna try to eliminate answers as well.
And I can also hear Mead's voice in my head. I think it was one of our last lectures. Yeah.
Okay. So the answer, enhancing oncogenic potential of rapidly differentiating cells. That doesn't sound right.
Yeah.
It's really specific too.
And there would be an enzyme doing that. An enzyme creating disorder like that doesn't make a whole lot of sense to me intrinsically.
Yeah. What about reversible inactivation of genes as cells terminally differentiate?
Okay, well, methylation is a covalent thing, so that doesn't sound reversible.
Right. And doesn't... yeah, I mean, I guess this is me. I have like one little voice in my head that says methylation turns on genes.
I don't know. I'm guessing, so you could be right.
Well, your answer doesn't say that it turns them off, so that's good. And then the other possible answer says stable silencing of DNA as cells terminally differentiate.
Which would be against what you're thinking, yeah.
I like your answer. Let's go for it. Please be right.
Nope.
Oh my gosh, we were so wrong. Oh, let's keep on just trying to talk while we figure out what the right answer is. There it is. Wow. Okay, stable silencing of DNA as cells terminally differentiate. So I was backwards.
I have no idea.
When you methylate, I guess. Okay, so here we go. Let's look at the hints. CpG methylation diminishes the oncogenic potential of cells.
Okay.
Okay, so now that we know that it silences them, that makes sense. CPG island methylation does not have a role in the differentiation of cells.
Maybe it's a different type of methylation, and I was just generalizing where I can't do that.
Okay. However, once cells are done differentiating, their DNA needs to be turned off or silenced. CPG methylation permanently or stably silences the DNA of cells that have completed the process of differentiation. Oh, so you use methylation to silence the parts of the genome that would be transcribed in other areas. Okay, next question, question number 6. What is the role of an activator? Displaces a repressor protein from an operon, allowing RNA polymerase to proceed in transcription, causes conformational changes in the DNA, bringing promoter regions into proximity with enhancer regions, binds to an operon and transcribes the RNA, or enhances interaction between RNA polymerase and the promoter. Okay, so role of an activator. Enhances interaction, RNA polymerase and promoter. That doesn't feel right. Binds to an operon and transcribes RNA. No, that would be an RNA polymerase. Okay. Causes conformational changes in the DNA, bringing promoter regions into proximity with enhancer regions. I have no idea. I wouldn't assume that, but I really don't know. So I'm going to keep them on the table. Or displaces a repressor protein from an operon, allowing RNA polymerase to proceed in transcription. That sounds right to me. What do you think?
I'm actually between two of the others that you previously mentioned.
Oh, no.
Causes conformational changes in the DNA, bringing promoter regions into proximity with enhancer regions. I guess not necessarily. I think that is what enhancers do.
So an enhancer is something that increases transcription, right?
Yeah, and I think...
And promoter turns it on.
So, like the TRP, the tryptophan operon, I think is normally... Oh. No, because that one's normally on. And
so...
Opposite. Never mind.
So if you're bringing promoter regions, which is a region of DNA, in proximity with enhancer regions, which is a region of DNA, I feel like that doesn't do anything.
Yeah, I think that enhancer is something that brings promoter regions closer to something. Yeah, I think I'm getting my definitions messed up. It's very late. Enhances interaction between RNA poly and the promoter. I think I might go with D.
Okay, I could totally understand that too. Activator, yeah. I mean, I'm stuck between that one and displaces a repressor, but I'm willing to go for it. Hit check, check. Nice work, we got it.
Okay.
All right, so activators, everybody, they enhance the interaction between RNA polymerase and the promoter, meaning that they activate transcription. There's so many little verbs for like different ways that you can just start it. But this one activates. It enhances the interaction between RNA polymerase and the promoter. All right, next question. So, what is the function of a spliceosome?
Oh, thank God.
It's one of my favorite enzymes.
Okay, go. One?
It's two of my favorite enzymes.
I'm being sassy. I think it's a complex of like 5, 6.
It might be.
Right, because there's U1 through 6. Actually, it's 5 because there's no U3.
Well, I had no idea.
Okay.
All right, so the possible answers are to cleave telomeres prior to transcription, to condense DNA into small manageable units, to cause conformational changes in DNA that are amenable to splicing, or to cleave introns from RNA and ligate the cut ends. So this one you just got to know. This is a good definition. Lucky for all you biology majors out there. Sorry for everybody else. But yes, the point of a spliceosome is to clip out those introns that get stuck when you translate mRNA before it becomes mature mRNA.
In eukaryotes.
Right, yes. Yeah, just in eukaryotes.
Yep.
Okay, next question. Which of the following is not a function of the 5' terminus of mRNA? Promotes ribosomal binding for translation, promotes polyadenylation of the 3' terminus, protects the mRNA from degradation, or regulates nuclear export of mRNA. Okay, 5' terminus. So, 5' terminus of the mRNA, I don't think has anything to do with nuclear export. I don't think it promotes ribosomal binding for translation. It might. But I think isn't that ATG?
Ribosomal binding.
Yeah. Well, either way, if we're just talking about the 5' terminus, the one that jumped out at me was protects the mRNA from degradation, because I know they put on that what is it, the guanine cap?
Yeah. So that is one that is true. And this question asks for not a function.
Oh my gosh, I just committed the second cardinal sin of the MCAT, and I
Yes,
forgot to
you
look
did.
at not.
But it's almost midnight, so we can be a little lenient with ourselves tonight.
Yes.
After a full day of lots of other schoolwork. And so I think it might actually, since 5', since we make it amino to carboxy. Oh, I'm sorry. Now I'm talking about peptides. Well, there we go. The one I went with that is not a function of the 5' terminus of mRNA would be promoting poly A addition on the 3' tail. I just
Yes.
don't know how the 5' and 3' ends would be related, so I'm just not sure.
Okay, I think that's actually completely accurate now that I know that that the question is not, and I'm gonna hit check. Yay! Got it
So,
right.
I didn't necessarily know that the one that you mentioned regulates nuclear export of mRNA, but since 5' would be the first end that we're making,
Right.
it wasn't like an outlandish consideration.
Yeah.
So even though you might not think that's the right one, don't immediately cross it out.
Gotcha. Yeah. Okay, next question. MicroRNA primarily aids in transcriptional and post-transcriptional regulation by which method? Okay, this is where you need to know more than just tRNA, rRNA, and mRNA. So, gene silencing through translational repression or target degradation, cleavage of telomeres from 5' termini, RNA editing via deamination of specific DNA sequences, or gene amplification through binding directly to critical DNA promoter regions. So you can't really reason through this. I'm going to try. So cleavage of telomeres from the 5' termini. If you're talking about microRNA, figure it's probably not doing something like cleaving telomeres. I guess it's possible because I think some RNAs do have endonuclease activity. But RNA editing via deamination of specific DNA sequences, just none of this is right. I guess really the way you got to just look at it is the exact definition, which is microRNA. The purpose of it is to kind of act as a post-transcriptional repressor. So my answer was gene silencing through translational repression. Ooh, translational repression. Oh, sorry. Yeah, you're stopping translation from happening by
post-transcriptionally.
Gotcha. Yeah, post-transcriptionally, which would be this complementary microRNA strand binding to one of the mature mRNA transcripts that gets sent out to a ribosome and preventing it from being translated.
So I didn't know what microRNA was, but I did get there by doing the MCAT reasoning stuff.
Awesome.
Cleavage of telomeres from the 5' termini didn't make any sense to me. I knew that would destabilize the RNA, and it was just not tracking. And so RNA editing via deamination of DNA, well, it says post-transcriptional, so I know I'm working with RNA.
That was good. Yeah.
And
Didn't see
then
that.
gene amplification. Well, gene, anytime I'm mentioning gene, I'm working with DNA. So critical DNA promoter regions, well, I'm working with RNA like they told me.
And that's pre-transcriptional at that point.
Yeah. So it was a guess. It was definitely a guess.
And we got it right. All right. So, final question. A proto-oncogene can be converted to an oncogene by all the following except increased repressor binding, chromosomal rearrangement, gene deletions, or gene amplification. Okay, so you were pretty quick with the trigger right there. I do not know exactly what I'm doing, so
Okay.
teach me something.
Okay, so do you know what a proto-oncogene is?
Not entirely.
So there's a couple of genes that if something were to happen in them in just the right way, they could be cancer causing.
Mm-hmm.
And so that is how we talk about those. And so I would know that chromosomal rearrangement is mutating DNA in ways that are probably never good. Gene deletions, that's a frame shift. That's a big deal. Gene amplification, well, if it's something that could cause cancer, I don't want to make more of it. And so increased repressor binding is the exact opposite of that, decreasing something that could potentially be dangerous in the right conditions.
Okay.
Yeah.
Yeah, so we're talking about cancer. Increasing repressor binding seems like the opposite of cancer. Well, except for maybe like P53.
I do not know that one.
P53, it's a cell cycle modulator. I'm pretty sure P53 gets released. Well, you know what? No, P53 would be upregulated, I suppose, in cancers. I think P53 gets released to tell the cell, yeah, you're good. Go from G1 to S phase, and then go from G2 to mitosis.
With no checkpoints.
Well,
Or just
P53
overriding.
is the checkpoint,
Ah.
and I think it gets upregulated like crazy in cancerous cells.
You would have more checkpoints in a cancerous cell?
No. It's not the checkpoint. It's the okay signal.
Oh, okay.
Yes. So,
I need to
hey, and
review
you're right.
cell cycle.
Yes, I do too. But I'd like to look at the hints of these real quick. So a proto-oncogene can become oncogenic by deletions in the gene or by chromosomal rearrangements. Okay, yeah, I've heard about that before. Any increase in protein expression can contribute to oncogenicity, and this can be caused by gene amplification. Okay, yeah. And then an increased repressor binding is unlikely to cause oncogenic conversion, as this will result in decreased protein generation. All right, that made sense. We got 80%. Not bad.
Would like to mention that on a lot of the science passages, they seem to really like the idea of asking you if such and such is messed up and this thing is increased, what will happen to some other resultant variable? And be very careful, like maybe highlight the relationships between such and such and the other dependent variables.
Oh, yeah.
Yeah, and so knowing these rules for genetic stuff is really good, but also being able to pick that out in given data is important.
Yeah, this one was pretty tough. It's also like relatively obscure content that doesn't stick with you that well, but when you look back at it, it's actually very important.
Yeah.
Unless you work in a lab or you're just totally down with gene control questions, I guess. All right, strong work, gang.